background of Elizabeth. Dr. Elizabeth Henaff is a computational biologist with an art practice whose research focuses on the ubiquitous and invisible microbial component of our environment. Please correct me, the pronunciation, Elizabeth. This inquiry has produced a body of work that ranges from scientific articles in peer-reviewed journals to projects with landscape architects to artworks shown nationally and internationally. Some recent artworks include commissions for the Storefront for Art and Architecture in New York City, the Detroit Science Gallery, and the Okayama Art Summit in Japan. Using New York City as a living laboratory, her research group at NYU Tandon School of Engineering investigates microbial metrics in urban environments with a focus on anthropogenic change. Research directions include engineering of green wall infrastructure, remediation of Superfund sites, and the impact of street-level flooding on urban microbial diversity. As you may already know, this week she's giving a biointeraction workshop at the Master in Design for Distributed Innovation. And today we will have the first hour of an open lecture as we look at how current reality unfolds through bio-human relationships and how this can create the conditions for life to emerge through innovation. So the stage is yours, Elizabeth.
Awesome. Thank you so much for the introduction. I'm looking forward to getting to know all of you better over the course of this week as we work together. So what I'm going to do now is for the next 45 minutes or an hour, I'm going to give a mile-high overview of why I think environmental microbiomes are interesting, crash course in biology, where we're going to talk about microbes as related to multicellular organisms, so as related to plants, as related to animals. We're going to talk a little bit about cell structure, and then that's going to be the first hour that we spend together. And then I have planned for you guys today a couple of different activities that we're going to do together, and we're going to dive deeper a little bit into specific types of microbiomes. So for the people on the internet who are watching, welcome for this first hour with a crash course in holobiont design and the multi-species city. So I am going to share my screen. There we go. I'm going to arrange it so I can see all of you. And let's see if this works. All right, can you see white text on a black background?
Yes, perfect.
Awesome. So as scientists, we often use instruments to mediate our relationship with the things that we want to study. Oftentimes we want to study things that we can't see with the naked eye. So we invent instruments to extend the capacity of our sensory systems. So for example, the first image here that you see on the left is a transmission electron microscope image. So that's an instrument that allows us to see what is very small. So what we see here are chromosomes, so DNA molecules that have condensed and are ready for cell division. Or the image in the middle, for example, is an X-ray. So that's an instrument that allows us to see the hard tissues inside the living body of an organism. So we are able to understand their structure and function in certain ways in a non-invasive manner. And the image that you see on the right is an image of a DNA sequence. So a readout, a digital readout of the molecular structure of a DNA molecule of an organism. And so this instrument, even though DNA sequencing is a process, because it requires many steps to get that information. You can also consider it as an instrument that allows you to perceive that information. And that's the instrument that I've been most interested in using for the last decade or more. And so this is an instrument that allows us to get a readout of the information storage molecule used as an information system of all living organisms that we can study to date. So we can do all sorts of interesting things with that instrument. We can point that instrument to ourselves, human beings. So the use of DNA sequencing has enabled the era of precision medicine. So say, for example, you are diagnosed with a cancer, your oncologist is likely going to prescribe a whole genome sequencing of, for example, an excised tumor and a healthy tissue. And by reading that information and comparing those two genomes, they might be able to see differences that allow them to guide your treatment plan. But we can also turn this instrument to the environment to help us understand better the environment and ecosystems that surround us. So I'd like for you to take a second to close your eyes and conjure a mental image of what you think of when you think of the environment. So just take a couple of seconds to do that. When I think of the environment, I might think of something like this. So a lush green landscape, plants and animals. This image is taken from the film or the animation Princess Mononoke by Studio Ghibli, which is one of my very favorite animation studios. But when we think of the environment, oftentimes we think of these lush green landscapes, maybe even landscapes that are far away from us, that do not resemble the landscapes that we inhabit. Sometimes a human being will make their way into our vision of the environment, but oftentimes not. I've asked this question a bunch of times now, and I would say it's a very small proportion of times that people envision human beings in their environment. We can talk about that later. When we gather as a small group. But what is not visible in this vision of the environment are the billions of microorganisms that inhabit even just a teaspoon of soil from this forest. And so what we're starting to be able to do with this instrument of DNA sequencing is understand the microorganisms, the invisible kind of biological component of our environments. Because up until the last couple of decades, what we knew about microorganisms was what we could tell from microorganisms that we could grow in a lab. So you take a sample somewhere, you swab it out in your petri dish, you put it in your incubator, and you see what grows. But what grows depends on a lot of things. It depends on what temperature your incubator is at. It depends on what kind of medium you use. It depends on whether that microbe is actually willing to grow in a condition that is not its native condition. So we call microorganisms that won't grow in a lab recalcitrant microorganisms, which I love because it makes them seem just kind of grumpy, but for good reasons. They don't have to grow in the lab. And so what's exciting about DNA sequencing is that it allows us to get a less biased vision of environmental microbiomes. I say less biased because all tools are biased in some way or another. But what helps reduce bias in this particular case is that we don't have to grow them to know something about them. So we can just take that teaspoon of soil, extract DNA directly from it, and sequence it, and get a broader picture of what's living there. And so what's emerging from this is that we're starting to understand that a lot of key characteristics or phenotypes of these multicellular organisms like we see here, plants, animals, are related not only to the genotype of that plant or animal, but also to their relationship with the symbiotic microorganisms with which they cohabit. So for example, it was shown that flowering time in plants can be altered by altering the microbial composition of the soil in which they're growing, which is kind of big news in the plant biology world, because flowering time was thought to be the canonically well understood, genetically determined pathway. Like plants have different life strategy decisions as to when they flower, maybe before the cold, after the cold, depending on daylight and time. So this was really thought to be like a internally programmed system. So it totally rocks the plant biology world when we realized that system actually is dependent not only on the genome, just the genotype of the plant, but also the genotype or composition of the microbes with which it's interacting. So this is the case also for mammals, including human beings. So we're getting really comfortable with the idea that the gut microbiome, for example, is really important for human health. But also those interactions can have much more kind of subtle effects on human health and well-being. And so we're finding that the synthesis of neurotransmitters in the brain, upon which depends proper brain function and mood, etc., are impacted by these gut microbiomes. So this is just a couple of examples of many, many examples that exist in the literature that show that physical characteristics or phenotypes of a multicellular organism, like a plant, like an animal, like a human being, like ourselves, are due not only to the genotype of that individual, but also to the genotype of the microorganisms with which we live in symbiosis, with which we interact. And so if we think of the genotype, so like our genome, our set of genes, as the definition of identity, of genetic identity, then that kind of begs us to broaden the notion of identity to include not only our own genetic information, but also the genetic information of the microorganisms with which we interact. And so Lynn Margulis, who's a evolutionary biologist and also feminist theorist, coined the term decades ago now of holobiont to describe that broadening of the notion of the individual. So the holobiont is a term that encompasses the notion of host in symbiont. And so she proposed that we should consider the boundaries of our identity or of ourselves as porous to be able to include these other organisms. And so for me, it's really interesting to kind of hold this particular idea of porous boundaries when we want to ask questions about humans inhabiting spaces, about the urban environments that we inhabit. So imagine we take, we have this instrument of DNA sequencing in hand, and we're like, I'm going to study my environment. But for a lot of us who live in cities, our environment doesn't look like a lush green landscape. It looks, something like this. So over half of the global population lives in cities at the moment, and that's projected to increase. And so our environments mostly look like this. And so imagine we had, our like DNA sequencing goggles on, and we were studying our environment. It might look, something like this. So we can understand the microorganisms that are associated with our built environment. We can understand the microorganisms that are associated with the human inhabitants, and everything in between. And so what's interesting to me is that we can, study the relationship of humans and our microbiome and the impact on human health, but also the human microbiome interacts with and is impacted by the environmental microbiome, and the design decisions of practitioners like architects, material scientists, city planners are sculpting this environmental microbiome. And so as such, oftentimes those impacts are unintentional. And so the design practitioners designed to optimize all sorts of metrics, but often not that biological metrics. So just let's think about what it means exactly to design for a metric, for example, for a second. So we have all sorts of choices available to us for the design of the built environment. And generally speaking, we have choices of materials and we have choices of shapes. And oftentimes these two parameters are chosen in order to optimize human comfort. And human comfort is measured in all sorts of ways. So we can measure things like temperature, for example. That's something that's easy to measure and to perceive in our environment. So this is an example of a built environment structure in which the materials and shapes are optimized for thermal comfort. So these are adobe houses in the southwest of the United States that are built with extremely thick walls and very small apertures to maintain cooler temperatures inside. We can also measure sound levels, and we can choose our materials and shapes in order to optimize acoustic comfort. So this is the design of a concert hall in which the shapes of the concert hall are meant to give an optimal acoustic experience to all of the audience independently of where they're sitting. So those are just a couple of things that we can measure about our environment, but we can measure a bunch of other things. We can measure light, for example. We can measure air quality. We can measure CO2 concentration. And we make all sorts of decisions for our built environment in order to optimize these things that are available to us to measure. But we can also, but what's emerging now is that now with this tool of DNA sequencing, we are able to develop a microbial metric of our spaces. And so the challenge, I think, to me in design for the next century is being able to appropriately use this microbial metric to fold that into our design practices. So in this case, it would be choosing materials and shapes, but optimizing for human microbe collaboration. So if we go back here to our idea of the urban microbiome, what we will be doing is choosing the materials and shapes of our built environment in order to select for a particular microbial population with the goal of some kind of advantageous or at least not disadvantageous interaction with the human microbiome. And so for that, we need to do a bunch of different things. We need to be able to measure these microbes, so understand who is there, so species classification, understand what they're doing, so what kind of metabolisms they encode. We need to be able to represent that information, which is particularly challenging when we're operating at such vastly different scales. So microbes are measured in microns. We measure our built environment at the scale of meters or kilometers. And it's actually very difficult to be able to represent information across those different scales. We might want to be able to alter these microorganisms or alter our built environment as a function of what we measure. In that sense, we want to be able to design for these organisms. So being able to, again, choose materials and shapes in order to select for specific populations. Ultimately, we want to be able to interact with these. If the goal is to be designing interactions, we want to be able to interact with them, which again is particularly challenging because these are organisms that we can't see. And so for that reason, we need to build instruments or extensions of our sensory systems, or maybe even build particular methods of observation to allow us to understand that. And so what I wanted to do today is just kind of give you a mile-high overview of examples of the ways that we do each of those things. And then we'll get into talking about microbes specifically and why they're interesting. So there's all sorts of ways that we can measure DNA. This is a particularly cool one. This is a portable USB DNA sequencer that you can use in the field. We've used it in the subway, but also it's been used in the field, for example, for epidemic surveillance. So with that type of measurement, what we do is we collect microorganisms, we extract their DNA and sequence it, and then we use that data to map against genomes of known microorganisms. And then what we can do is we can, for example, classify these microorganisms. So if we have one of our DNA sequences in our kind of mystery sample map genome that is already known in the database, we can say that that species is there. And depending on how much of our DNA maps to that particular genome, then we can say that we have more or less of that original microbe. There's tons of ways that we can represent this information. This is one that study that I participated in a number of years ago now, in which we studied, sampled all the subway stations in New York City, and worked with the Wall Street Journal to make an interactive visualization so that you could see which microbes you were interacting with on your commute. This is definitely not the first microbial map to be made of a city. The first one that I know was made in the mid 1800s by Dr. John Snow of London during a cholera outbreak. And so what this doctor did, so this was before there was an understanding of microbial transmission mechanisms of disease. And so what this doctor did is took a map and colored in all of the homes in which there was a case of cholera, and using this data visualization was able to identify that what those houses had in common were the use of particular wells, of water sources. And so upon closing access to those wells, they were able to curtail the epidemic. We can think of designing for microorganisms, so even designing materials for microorganisms. Oftentimes our materials are designed with antimicrobial properties for all sorts of reasons, for material degradation concerns, also because we just really like for our indoor environments to be super asepticized. So we put, antimicrobial components in paint and our clothing. But this is an example of a bioreceptive cement that was developed in a lab in Barcelona, which was designed to be able to host microbial populations, and lichen in particular, on the surface of those materials without affecting the structural composition or of the material. And then ultimately we have tools to interact with these microorganisms. And so here on the right, you can see this tool that we're all overly familiar with over the last few years, which is the swab that we have all had uncomfortably stuck up our nose way too many times. But that's a tool to collect microbial information. This is a air quality sensor that I saw in the subway one day. So this is also a way of collecting microbial information. But these are two kind of technological devices that belong to the modern era, not just the modern era, but like the Western science concept of the modern era. But it is definitely not a new thing that we have tools to interact with microbes. So for example, winemaking is a microbial interaction process that is as old as human memory, probably. And there's also different ritual practices that we can understand also as mediating interactions with microbiomes. So this is, there's a hypothesis that bioprecipitation is bioprecipitation is the mechanism behind why rain dances work. And we're going to talk about that a lot more in our workshop later today. But we also like to invent all sorts of other kind of collaborative interactions with microbes in the form of probiotics. And so this is an example of, I think, the only probiotic skincare that exists, to my knowledge. We're really comfortable with taking probiotics for our gut, but this is a probiotic for the skin. So as a summary, these are all the different kind of steps that need to happen to be able to think about designing for microorganisms. And so this is an idea that we're going to keep coming back to over the course of the week, is how do we measure things? How do we represent them? What does that mean for designing for them? And also, what does it mean to design tools to interact with them? Because unfortunately, We do always need tools to interact with these microorganisms because we're not able to perceive them with our naked eye in most cases. In some cases we can. So we don't have the superpowers of Moyashimon. This is a Japanese manga and anime in which the character has this superpower of they're able to see microorganisms in all environments and also hear them and talk to them. But we unfortunately don't have this superpower innate, but I think that there's things that we can do to train our way of knowing and to train our way of seeing and thinking that would enable us to be able to see the world in that way. And I think it's particularly challenging because we live at vastly different scales, orders of magnitude different. And so just to kind of contextualize this for you, because I think that we're not really used to thinking of orders of magnitude within the small. So anything that's invisible to us then just gets put into this one bag of like invisible and small. But there's actually vast orders of magnitude within the small. So if you think of the human body being measured in the scale of meters, approximately 2 meters, human cells are measured in hundreds of micrometers. So that's, 1,000,000 times smaller. Bacterial cells are then again 100 times smaller than a human cell. And then the width of a DNA molecule is another 1,000 times smaller than the bacterial cell. So this is something that we're also going to be trying to develop together over the course of the week, is this kind of mental model or mental image of what does the world look like at a microscopic scale, and how even at a microscopic scale, you need to imagine interactions between entities that are themselves vastly different in size. How are we doing on time? I think we're doing all right. So what I wanted to do now is give kind of a mile-high view crash course in why I think microbes are awesome. Basically, that's the summary. But to talk about microorganisms in from a few different perspectives. So we're going to do a brief overview of cell structure. We're going to talk about how microbes live in communities. They don't live as individual organisms. And then we're just going to run through a bunch of examples of microbes in different environments, in soil, interacting with plants, interacting with mammals, and also the built environment. So back to biology 101. This is a cell. On the right here, you see a eukaryotic cell. So plants, animals, like ourselves, humans, we are all eukaryotes. And on the left here, you see a prokaryotic cell. So there's a couple of major differences that are important between these two cells. One is eukaryotic cells are a lot bigger, like 100 times bigger on average, which is not exactly represented to scale in this drawing. What allows them to be bigger is the fact that eukaryotic cells have subcompartments within them. So I don't know, can you see my mouse right now? Yeah, awesome. So what allows eukaryotic cells to be bigger is the fact that they have these subcompartments. So they're able to kind of compartmentalize and therefore specialize different cellular functions in different places of the cell, which allows them to get bigger. Because if you think of molecular interactions as being a function of chance, of Brownian motion of these molecules in the cell, and you're like, okay, substrate A really needs to meet enzyme B, but it's only going to happen by chance of those molecules just bouncing around the cell. If you have a very, very large cell, then those chance interactions are not going to happen as frequently as you want for them to. But if you make compartments in your cell and you compartmentalize substrate A and enzyme B that you want to work together, then you are increasing the chance that that happens. And because now you have compartments for kind of everything, you have compartments for energy and compartments for waste and compartments for protein synthesis and so on and so forth, then you can make a much bigger cell. On the other hand, prokaryotic cells only have one compartment. So everything happens in that one space. You only have one chromosome, one main chromosome, as opposed to, up to say 20 chromosomes or 22 chromosomes for humans. So prokaryotic cells are a lot simpler in that sense, but a lot more kind of agile in another sense. So for example, prokaryotic cells are able to just absorb DNA from their environment and start using it. But a eukaryotic cell is not able to do that, partially because of the compartmentalization, and also partially just because of the mechanisms of DNA usage. I like to think of an analogy of a cell functioning like a construction site. So we're going to talk a lot about DNA as a way of knowing organisms. And so it's important to remember that DNA is an information storage, and it's not a functioning molecule. So if you think of a construction site, here on the left, you have your machines, like these cranes and trucks, and then you have your materials, like your bricks and cinder blocks and stuff. And the function of these machines and how they're going to assemble those materials is dictated by your blueprint. And so you maybe store your blueprint somewhere really safe, like this little air-conditioned office that's over here. And you're going to just use your blueprint, maybe make copies of your blueprint to take to the part of the construction site where you're going to use that information. So the blueprint tells you what you're going to do with your machines and how you're going to assemble your three-dimensional structure, both in structure and in function. And similarly, DNA in a eukaryotic cell, so again, this is the cell which has compartments, is stored in its own little compartment. And that DNA, that information in that DNA, we're going to make a copy of just what we need, so one gene at a time, and we're going to take that into the part of the cell where we do protein translation, and that information is going to be used to assemble materials in the cell. So assemble proteins and enzymes that have a particular function, and so that's going to give you the kind of three-dimensional structure of your cell, both in structure and in function. So just keep that in mind as we're talking for the rest of the week, is that when we talk about DNA, this is talking about information storage that then leads to three-dimensional structure and function in like a physical space. So prokaryotic cells, microbial cells, and eukaryotic cells, so plants and animals, exist at vastly different scales. So here we see an electron microscope image of these little rod guys that are actually assemblages of multiple microbial cells with an animal cell compared to it. So you can see that there's, roughly, and these exist in three-dimensional space, right? So there's like a 1 to 100 or a 1 to 1,000 relationship in size between these two cell types. So microbes exist in communities most times. They like to hang out with each other, sometimes within the same species and sometimes within different species. So one of the most obvious form of microbial communities is biofilms. So this is something that is observable to the naked eye. So for example, if you're growing a kombucha culture, that's a biofilm. If you forget like a can of soup in the fridge for a really long time, and there's something that starts growing at the top, that's also a biofilm. You see them happen in contaminated environments also. So these are assemblages of microorganisms that create a matrix for themselves that in a certain sense creates a body that they can inhabit. So it allows them to distribute themselves in three-dimensional space and kind of specialize in functions just like our three-dimensional bodies do. Then they also have decision-making strategies for when they need to stop being in a biofilm, when the biofilm has reached its capacity, and when they need to leave. So that's what this paper is talking about here. Just to give you a visual image, these are really gorgeous fluorescence in situ hybridization electron microscope pictures of different microbial species that inhabit dental plaque. And so what you can see here in some of these images is that there's a three-dimensional structure to these. So you can see here, so the different colors correspond to different species. So in this here, you can see that the green species is on the outside, the purple species is on the inside. And this is actually a difference between being aerobic and anaerobic. So the species on the outside consumes all of the oxygen available and creates an anaerobic environment for the species on the inside. So you can see that these assemblages are creating three-dimensional structures that can actually have really vastly different kind of microclimates that these microbes are going to inhabit. Another one of my favorite examples of biofilms are lichen, which are observable also to the naked eye. And these are actually assemblages of both fungi and algae, photosynthetic algae. So now I want to go through a few examples of microbes in different environments. So microbes in soil are really kind of key intrinsic components of soil composition, and they respond to their environment. And so this is a paper that showed that you can actually use microbial communities as quantitative sensors for abiotic or non-living characteristics in the soil. So for example, like if you have a high pH, then you're going to have microbes that correspond to high pH. Or if you have heavy metals, then you're going to have microbes who are resistant to heavy metals. So actually by measuring the microbial community, you can know things about the geochemical environment without actually having to measure the geochemical environment. Microbes also respond to dynamic changes in their environment. So there's a whole series of really awesome papers following the Deepwater Horizon oil spill in the Gulf of Mexico a number of years ago now, that showed that the endogenous microbial community responded very quickly to the oil spill and changed their dynamics to increase or not. So yeah, to increase the proportion of microbes that are able to digest petrochemicals. Sadly, the dispersal agents, so the chemical dispersal agents that were used as a cleanup strategy to disperse the oil spills, actually inhibit the natural degradation of the petrochemicals by the endogenous marine microorganisms. So microorganisms are also very key in plant growth and development, and constitute these really gorgeous and amazing kind of porous continuums between the plant body and the roots and the soil structure. This is called the rhizosphere. So also as you're building your three-dimensional imaginary of the small, you can imagine these rhizospheres that are surrounding all of the roots of the plants that you can observe. Some plants have very explicit collaborations with microorganisms. So these are plants like soybeans, for example, that will have a very explicit relationship with nitrogen fixing microorganisms. And so the relationship is that the plant is able to perceive signals from nitrogen fixing bacteria. They grow a structure in their root, so kind of these bulbous structures that you see here, specifically to house these nitrogen fixing bacteria. The collaboration is that the plants then have access to bioavailable nitrogen for their growth and development, and they provide the bacteria with sugar. So this is a very explicit and kind of collaborative relationship between these organisms. And there's a bunch of examples about plants and microbes that are really cool. One is that grapevine-associated microbiota come from the soil. So the actual fermentation, like fermentation of wine, when people use a natural fermentation process, is directly dependent on what's going on in the soil at the moment. And so we talk a lot about terroir as a concept of, like, in particular in food, of like food and taste being associated to a particular place. And we often think of terroir as being a function of climate, so temperature and rainfall. But what's emerging is that really terroir is related to the microbial environment of a particular place. That's the case for wine, that's the case for cheese. Also, another really cool example is that, we use plants as medicine in all sorts of different ways all across the world and over time. And so it's emerging that some of the medicinal qualities of certain plants might not be produced by the plant themselves, but by microorganisms that inhabit the plant structure itself, so that are producing these secondary metabolites or small molecules that then we use for medicinal purposes. Microbes also collaborate with mammals in all sorts of ways. So this is a really great paper that describes inheritance of microbiomes in mammalian generations and their influence on animal development. And so there's actually two modes of inheritance. There's actually like internal inheritance during gestation, and then there's also inheritance from the mother's body. So in the case of humans, that's inoculation of the skin microbiome for the infant as the infant exits the vaginal canal, and also gut microbiome from breastfeeding. What's really cool is that the mother's vaginal microbiome will change from looking like a vaginal microbiome, which is very specific, to looking like a skin microbiome a few days before birth. So it'll actually alter its composition a few days before the baby is born, in order to be able to inoculate the infant with a fresh skin microbiome. We talked about this paper already earlier about how microorganisms are key in the signaling pathway that determines synthesis of neurotransmitters in the brain. And also gut microbiomes are, definitely emerging as being a very important component of health and well-being. And so next, the built environment. So we talked about microbes in our transportation systems. So if you happen to be in New York City in 2015, you can figure out which microbes you're interacting with as you're taking the subway. This paper did a similar study in Boston, and what was really interesting is that they went a little bit further and they started talking about the different materials in the subway system and how those materials would select for different microbial populations. So they looked at the difference between a stainless steel pole versus a plastic strap versus a cloth seat in the subway. And so this is really starting to get to the aspect of design where, material choices that were made for this aspect of the built environment that were probably optimized for all sorts of other things like durability and material properties like flexibility, and is it easy to, clean or something like that. But they were actually also selecting for this microbial metric. You can also look at this in indoor spaces. So this is one of my favorite papers that talks about biogeography of indoor spaces. And that establishes kind of a theoretical framework of a way of describing connectivity of indoor spaces and how the connectivity of indoor spaces influences the distribution of microorganisms in those indoor spaces. So here, they describe these different connectivity methods. So, radial, meaning like a space that is connected to many other spaces or connected in a grid, or linear. And then they start to create these relationships about the design of the architectural design of indoor spaces and microbial populations. Obviously, microbes can also have very detrimental effects on our built environment. And microbial deterioration of materials is a real thing. There's a reason why we do all sorts of things to treat our materials so that they are not microbially deteriorated. And this is an older paper, but I think it's really great because it has a really good list of types of species, the types of degradations that they perform, and the types of materials that those are related to. So this is a really good reference when, you know, we try to like wax a little too poetic about like bioreceptive urban environments and everything, but we can also kind of check ourselves and make sure that, you know, our buildings are going to still be standing in 20 years. We talked about this already. So developing materials specifically with those two concerns in mind. So the idea of being able to host diverse microbial populations and also being able to maintain structural properties. So what we did today is we did like crash course, mile high view of, you know, what is a microbe? How can we imagine it in three-dimensional space in relationship to ourselves and to our bodies? And also, how do microbes exist in all these different environments? With ultimately the idea that we know that human health and well-being is related to our own microbiome, and that our own microbiome is impacted by and interacts with the environmental microbiome. And we also know that exposure to diverse microbial environments is really important for human health and well-being, in particular in early stages of development. However, a lot of the design decisions that we make for the built environment, so the way that we design our HVAC systems, the fact that our office buildings don't have windows that we can open, the antimicrobial materials that we use in our spaces, a lot of the design decisions that we do are actually degrading that diversity, so leading to a lower diversity of our spaces. So the challenge that I see is, how do we encourage microbial diversity in urban environments while still being conscious of all of the constraints, the material and physical constraints of the built environment? And so that's something that we're going to be talking about over the next week together at a bunch of different scales. And so finally, I would love, you know, to just leave you guys with this idea of the fact that that the instruments that we use to interact with these microbes or to perceive or measure these microbes matter. We don't have direct X-ray vision to be able to see the microorganisms around us. So all of our experiences are going to be mediated by an instrument. And we would like to think of instruments as being these kind of unbiased extensions of our sensory system, but they're not. We build instruments with a particular goal in mind. And so therefore they contain and they manifest all of our hopes and dreams and biases and preconceived ideas about the thing that we're measuring. So for example, imagine if you take a look at this image on the left, the swab, this is an instrument that is designed for interaction with a microbial community. So it's an instrument with which we're going to collect biological material. And this instrument has a really strong like affective quality to it. It makes us think of hospitals and disease and contamination for perfectly good reasons, because it is designed as a clinical instrument. When we were doing the subway study and using these swabs to collect, you know, biological material in the subway, this was definitely pre-pandemic. The teams, the swabbing teams, had all sorts of really strong reactions from subway passengers. So people were afraid that there was an epidemic, that there were people that you were monitoring for an epidemic. Some of the team was accused of implanting HIV in the subway, so accused of bioterrorism. And that's just a couple of examples of these really strong reactions to the tool. And so you can see that the tool really matters. The tool is the thing that we have the relationship to. It's not the microbes that we have the relationship to. Because the image on the right here is doing exactly the same thing. So grabbing a handful of soil is also taking a microbial sample. but we have very different feelings about it. So the image on the right gives us feelings of, you know, groundedness and earthiness and connection. It feels great, right? You can just imagine how that soil feels in your hands and it just makes you feel good. But ultimately those two images are doing exactly the same thing, but those are two tools that have completely different affective relationships with. So the tools really matter. And so that's ultimately where we're going to go, where land at the end of this week is thinking about designing collaborative tools with microorganisms. So just as a summary, what we're going to be doing this week is the first day. So we're going to be talking about microbes at a planetary scale. Tomorrow, we're going to be talking about microbes and anthropogenic contamination. The day after that, we're going to be talking about microbial diversity and environmental justice. Then we're going to be talking about design methodologies across scales. And then Friday, we're going to do a design sprint together. And in between all of that, there's all sorts of activities and stuff, both in class and also at home that I've planned for you guys. And that's what I had. Oops, that's for us later. I'll stop sharing my screen.
Amazing. I'll